My name is Meagan Olsen, and I am an undergraduate student in the lab of Dr. Shannon Servos at the University of Arkansas. My research is centered around the functionalization of spherical gold nanoparticles with peptoids, which can bind to viral envelope proteins. So peptoids are poly-N-substituted glycines, which is basically just saying that they're kind of like synthetic peptides with a few modifications. Because peptoids have their side chains on the backbone nitrogens instead of the carbons, they're not able to be degraded by proteolytic enzymes that can break down peptides, which becomes important when working with biological samples. The idea is that by coating gold nanoparticles with peptoids, we can develop electrochemical sensors with improved response times, specificity, and accuracy. This would allow us to develop point-of-care diagnostics for viral species that are currently hard to distinguish between using traditional methods. So for this work, we focused on a proof of concept peptoid capable of binding to green fluorescent protein, or GFP, to help with our analysis process. So nanoparticle functionalization is complicated by the fact that adding peptoid leads to aggregation. This aggregation is accompanied by a shift in solution color from pink to purple or clear, and it can be monitored both visually or by looking at how the peak location shifts and broadens in the UV-Vis spectra. There are a variety of factors that can impact this aggregation speed. So peptoids with different structures tend to have different rates of aggregation. The 4 samples shown here all use different peptoids and show different levels of aggregation after 2 hours. Using the proof of concept peptoid showed a second ago, we can see that aggregation can be increased by using an acidic pH, mixing the samples, or by simply adding more peptoid to the solution. Addition of phosphate buffered saline can also lead to rapid aggregation because there are salts present that disrupt the stabilizing citrate layer on the outside of the gold nanoparticles. Disruption of the stabilizing layer also seems to lead to nanoparticle adsorption or sticking to the centrifuge tube walls, making it hard to resuspend them later. Tubes coated with biological blocking buffers like casein can be used to minimize this particular issue. So to actually prevent aggregation during functionalization, we employed an intermediate monolayer of Tween 20, which is a bulky surfactant, and that can be used to sterically stabilize the nanoparticles, kind of like how security guards physically prevent people from getting too close to a band during a concert. This Tween layer replaces the electrostatically stabilizing citrate layer, which kept the gold nanoparticles apart because like charges repel each other. By making this exchange, the peptoids can then replace the Tween molecules instead of the citrates, allowing us to functionalize and isolate our nanoparticles without any of that aggregation, which can lead to problems in its functionality. To actually confirm that the peptoid bound to the gold nanoparticles and retained its binding capabilities, we took the functionalized nanoparticles and incubated them with green fluorescent protein and then isolated them. So the isolated gold nanoparticles do demonstrate an appreciable fluorescence after centrifugation and resuspension, and that can be seen here, which does at least somewhat indicate that the peptoid both bound to the nanoparticle and retained its ability to bind to GFP. That being said, the fact that the nanoparticle surfaces are so highly curved can lead to changes in protein conformation, which may have altered GFP's ability to fluoresce. There are therefore some uncertainty in these results, and we're working on potentially minimizing that uncertainty by applying another detection method like gravimetric measurements with a quartz crystal microbalance. Nanoparticle size also has an impact on the functionalization process. So we looked at both 10 nanometer and 40 nanometer spherical gold nanoparticles, prepared those the same ways, and they gave relatively similar functionalization results. However, the 40 nanometer particles were less likely to adhere to the centrifuge tubes, and they were not as impacted by the addition of methanol, which does seem to promote aggregation of the functionalized nanoparticles, more so for the 10 nanometer particles than the 40 nanometer ones. As noted previously, phosphate buffered saline, PBS, can promote aggregation of the unfunctionalized nanoparticles, while methanol seems to have little to no impact. We are still in the process of investigating this particular phenomenon. So over the course of this research, we determined a method to coat gold nanoparticles with peptoid without losing peptoid functionality. I'm currently working to extend this method to another peptoid of interest designed to bind to a viral envelope protein. We can then incorporate the nanoparticles and peptoids into an electrochemical sensor, incorporate those into an electrode, and hopefully improve point-of-care viral diagnostics through a relatively simple, specific, and rapid method. Thank you.